Today we are going to learn about the lymphatics and lymphoid tissues. Coming to the introductory remarks, when the circulating blood which reaches the capillaries, part of its fluid contents that passes into the surrounding tissues as tissue fluid. Most of this fluid re-enters the capillaries at their venous end. Some of it, however, returned into the circulation through a separate system of lymphatic vessels. Usually we call it as lymphatics. The fluid passing through the lymphatic vessels, it is called as lymph. The smallest lymphatic vessels are the lymphatic capillaries that join together to form large lymphatic vessels. The largest lymphatic vessel in the body is thoracic duct. It drains lymph from the greater part of the body. The thoracic duct, which ends by joining with the left subclavian vein at the junction with the internal jugular vein. On the right side, there enters the right lymphatic duct that has the similar termination. Scattered along the course of lymphatic vessels, there are numerous small bean-shaped structures. They are called as lymph nodes that are usually present in groups. The lymph nodes are masses of lymphoid tissue. The lymph nodes act as filters, remove the bacteria and other particulates matter from lymph. Lymphocytes are added to the lymph in these nodes. Each group of lymph nodes has a specific area of drainage. For the location of various groups of lymph nodes and the area of the body drained by them, we learned from gross anatomy textbooks. Aggregations of lymphoid tissue are also found at various other sites. Two organs, the thymus and the spleen, are most entirely made up of lymphoid tissues. The prominent aggregations of lymphoid tissue are present in close relationship to the lining epithelium of the gut. Such aggregations present in the region of the pharynx, which is consist as the Palatine tonsils. Isolated nodules of lymphoid tissue and large aggregations are called as paired patches that are present in the mucosa and the submucosa of small intestine. The lymph is transudate from blood and it contains the small proteins as in plasma, but in smaller amount and somewhat different properties. Suspended in the lymph, there are cells that are chiefly lymphocytes. Most of these lymphocytes are added to the lymph as it passes through the lymph nodes, but some are derived from the tissue drained by the nodes. The lymph capillaries beings blindly in tissues where they form a network. The structure of lymph capillaries is basically similar to that of the blood capillaries, but is adopted from much greater permeability. There is an inner lining of endothelium. The basal lamina is absent or poorly developed. The pericyte or connective tissues are not present around the capillary. As compared to the blood capillaries, much larger molecules can pass through the wall of lymph capillaries. These include colloid materials, fat droplets, and particulate matter such as bacteria. It is believed that these substances passes into lymph capillaries through gaps between endothelial cells lining the capillary. Now we are going to learn about the lymph nodes. Each lymph node consists of a connective tissue framework and numerous lymphocytes and other cells. The entire node is a bean-shaped structure, and the concavity constituting a hilum through which the blood vessels enters and exits from the nodes. Several lymph vessels enters the node on this convex aspects. Usually a single lymph vessel leaves the node through its hilum. When we take a section of lymph node, it is seen that the node has an outer zone that contains densely packed lymphocytes and therefore stains darkly. In this part, it is called as the cortex. The cortex does not extend into the hilum. It is surrounded by the cortex. There is a lightly stained zone in which lymphocytes are fewer. This area it is called as the medulla. Within the cortex, there are several rounded areas that are called as lymphatic follicles or lymphatic nodules. Each nodule has a pale staining germinal center and that's surrounded by a zone of densely packed lymphocytes. Within the medulla, the lymphocytes are arranged in the form of branching anastomosing cords. When we are closely observing the peripheral aspect of the lymph node, we can see it is surrounded by a capsule. The capsule, which consists of mainly collagen fibers, some elastic fibers and some smooth muscles may be present there. A number of septa or trabeculae which extends into the node from the capsule and that divides the node into different lobules. The hilum, which is occupied by a mass of dense fibrous tissues. Some functions of lymph nodes. They are the centers of lymphocyte production. Both B lymphocyte and T lymphocytes are produced here by multiplications of pre-existing lymphocytes. These lymphocytes passes into the lymph and thus that reaches the bloodstream. The bacteria and other particulate matters are removed from the lymph through the phagocytosis by macrophages. Antigen thus carried into these cells are presented to the lymphocytes, stimulating their proliferations. In this way, lymph nodes play an important role in the immune response to the antigens. Finally, the plasma cells that produce antibodies against invading antigens, while T lymphocytes attack cells that are foreign to the host body. And some clinical importance of the lymph nodes. The infections in any part of the body that can lead to enlargement and inflammations of the lymph nodes, draining the area. The inflammations of lymph nodes are called as lymphadenitis. The carcinoma usually spreads from its primary sites either by growth of malignant cells along the lymph vessels or by loose cancer cells passing through the lymph to the nodes into which the area where they drains. This leads the enlargement of the lymph nodes of this region. The examination of lymph nodes giving valuable information about the spread of cancer. In surgical excision of cancer lymph nodes, draining the region are usually removed. Next we are going to learn about the spleen. The spleen is the largest lymphoid organ of the body. The surface of the spleen is covered by a layer of peritoneum. Deep to the serous layer, the organ is covered by a capsule. Trabeculae that arises from the capsule that extends into the substance of the hilum. As they do so, the trabeculae divides the gland into small divisions. That forms a network. The capsule and the trabeculae are made up of fibrous tissues in which elastic fibers are abundant. The spaces between the trabeculae are provided by a network of reticular fibers that embedded in the amorphous matrix. The fibroblast and the macrophages are also present in relation to the reticulum. The intrinsics of the reticulum are provided by lymphocytes, blood vessels and blood cells and by macrophages. The circulations through the spleen. On reaching the hilum of the spleen, the splenic artery which divides into about 5 branches that enters the organ independently. Each branches divides and subdivides as it travels through the trabecular network. Arterioles arises from these networks, leaves the trabeculae to pass into its intertrabecular spaces. For some distance, each arteriole is surrounded by dense sheets of lymphocytes. These lymphocytes constitute the white pulp of the spleen. The arterioles then divides into number of straight vessels that are called as penicilli. Each of the penicilli shows a localized thickening of its wall. That is called as ellipsoids. The ellipsoid constitute the concentric laminae formed by the aggregations of fibroblasts and the macrophages. The lumen of each penicillus is much narrowed at the ellipsoids. Distant to the ellipsoid, the vessels dilates to form ampulla, the wall of which become continuous with the reticular frameworks. As a result, the blood flows into the space lined by reticular cells, coming into the direct contact with the lymphocytes present there. The part of the splenic tissues which is infiltrated with the blood in this way. This is called as the red pulp. The circulation in the red pulp of the spleen is thus an open circulation. Next, the white pulp. We have seen that the white pulp is made up of lymphocytes that surrounds the arterioles. As a result, it is in the form of cord-like aggregations of lymphocytes that follow the branching patterns of the arterioles. The cords appears to be circular in transverse section. At place, the cords are thicker than elsewhere and contain lymphatic nodules similar to these seen in the lymph nodes. These nodes are called as Malpighian bodies. Each nodule has a germinal center and a surrounding cuff of densely packed lymphocytes. The nodules are easily distinguished from these of the lymph nodes because of the presence of the arterioles in each of them. The arterioles is placed eccentrically at the margin of germinal center, and that arterioles called as the central artery. The functional significance of the white pulp is similar to that of the cortical tissues of the lymph nodes. Most of the lymphocytes in white pulp are T lymphocytes. The lymphatic nodules of white pulp are aggregations of B lymphocytes. The germinal centers are area where B lymphocytes are dividing. The red pulp is like a sponge. It is permeated by spaces lined by reticular cells. The interval between the spaces are filled by B lymphocytes as well as T lymphocytes, macrophages and blood cells. These cells appears to be arranged as a cord that is called as the splenic cords. The cords forms a network. The zone of the red pulp immediately surrounding the white pulp is the marginal zone. This zone has a rich network of sinusoids. Numerous antigen presenting cells are found close to the sinusoids. This region seems to be specialized for bringing antigen confined to circulating blood into contact with the lymphocytes in the spleen so that an appropriate immune response can be started against the antigens. Functions of spleen. Like other lymphoid tissues, the spleen is the center where both B lymphocytes and T lymphocytes multiply and play an important role in immune response. The spleen is the only site where an immune response can be started against the antigen present in the circulating blood. The spleen contains the large aggregations of macrophages of mononuclear phagocyte system. In the spleen, the main function of these cells is the destruction of red blood corpuscles that have completed their useful lives. In fetal life, the spleen is a center for production of all blood cells. In later life, only the lymphocytes are produced here. The spleen is often regarded as a store of blood that can be thrown into the circulation when required. This function is much less important in man than in some other species. Some clinical correlations. In conditions calling for increased production of lymphocytes, it is called as leukemia, or the conditions in which there is increased phagocytosis by macrophages, and in conditions involving increased destructions of erythrocytes, there may be enlargement of the spleen. This condition it is called as splenomegaly. Next, the thymus. The thymus is an organ that is a hazy entity for most students. This is because of the fact that the organ is not usually seen in dissection hall cadavers, because the organ is a rapid autolysis after death. The organ is also not accessible for clinical examinations. At birth, the thymus weighs about 10 to 15 grams. The weight increases to 30 to 40 grams at the time of puberty. Subsequently, much of the organ is replaced by fat. However, the thymus is believed to produce T lymphocytes throughout the life. The thymus consists of right and left lobes that are joined together by fibrous tissues. Each lobe has a connective tissue capsule. The connective tissue septa passing inward from the capsule incompletely subdivide the lobe into large number of lobules. Each lobule is about 2 mm in diameter. It has an outer cortex and an inner medulla. Both the cortex and medulla contain the cells of two distinct lineages. The medulla of the adjoining lobules is continuous. The thymus has a rich blood supply. It does not receive any lymph vessels but gives off efferent vessels. Epithelial cells or epitheliocytes. Embryologically, these cells are derived from endoderm lining the third pharyngeal pouch. The cells lose all contact with the pharyngeal wall. In the fetus, their epithelial organs is obvious. Later they become flattened and may branch. The cells joins to form sheath that covers the internal surface of the capsule, the surface of the septa and the surface of blood vessels. The sheath of the epithelial cells presents deep to the capsule, around septa and around the blood vessels, form an effective blood thymus barrier. That barrier that prevents the antigen from reaching the lymphocytes present in the thymus. Epitheliocytes also promote the differentiation and the proliferation of T lymphocytes. The cortical epitheliocytes are also described as thymic nerve cells. They destroy lymphocytes that react against the self antigens. Lymphocytes of the thymus or thymocytes. In the cortex of each lobule of the thymus, the reticulum formed by epithelial cells is densely packed with lymphocytes. Stem cells formed in the bone marrow travel to the thymus. Here they come to lie in the superficial part of the cortex and divided rapidly to form small lymphocytes. Lymphatic nodules are not present in the normal thymus. The medulla of the thymus also contain lymphocytes, but these are less densely packed than the cortex. As a result, the epithelial reticulum is more obvious in the medulla than the cortex. The macrophages. Apart from epithelial cells and lymphocytes, the thymus contains a fair number of macrophages. They are placed subjacent to the capsule at the cortico-medullary junction and the medulla. The subscapular macrophages are highly phagocytic. Deeper layer macrophages are dendritic cells. The first picture which showing the macrophages. Next, the corpuscles of Hussels. These are small rounded structures present in the medulla of thymus. Each corpuscle has a central core formed by epithelial cells that have undergone degeneration. These cells ultimately form a pink staining hyaline mass. Around this mass, there is a wall formed by concentrically arranged epithelial cells. These cells are stained bright pink with hematoxylin and eosin. The central mass of the corpuscles may also contain degenerating macrophages. The second picture which showing the Hussels corpuscles. The main function of the thymus is which helps to the formation of lymphocytes. It is called as lymphopoiesis. The proliferations of the T lymphocytes and their conversions into cells capable to reacting to an antigens probably takes place under the influence of hormone that produced by epithelial cells of the thymus. Myasthenia gravis is now considered to be disturbance of the immune system. There is some proteins to which acetylcholine released at motor end plates gets attached. The myasthenia gravis antibodies are produced against these proteins, rendering them ineffective. Myasthenia gravis is an example of the condition which the immune system being to react against one of the body own proteins. Such conditions are referred as autoimmune disease. Finally, the palatine tonsil. Each palatine tonsils, which consist of diffuse lymphoid tissue in which lymphatic nodules are present. The lymphoid tissue is covered by stratified squamous epithelium continuous with that of the mouth and pharynx. The epithelium extends into the substance of the tonsil to form several tonsillar crypts. The numerous mucous glands opens into that crypts. The lumen of the crypts usually contains some lymphocytes that have traveled into it through the epithelium. Desquamated epithelial cells and bacteria are also frequently present in the lumen of the crypt of palatine tonsils. The palatine tonsils are often infected, and that condition it is called as tonsillitis. This common cause of the sore throat. The frequent infections can lead to considerable enlargement of the palatine tonsils, especially in children. Such enlarged tonsil may become a focus of infection, and their surgical removal, the process it is called as tonsillectomy, may then become necessary.